Welcome back! Today I am going to go over a brief introduction to genetics as well as Mendel's experiments and the laws of probability. So what is genetics? Genetics is a study of heredity. Gregor Mendel set the framework for genetics long before chromosomes or genes had been identified, at a time when meiosis was not well understood. Mendel selected a simple biological system and conducted methodical quantitative analyses using large sample sizes. Because of Mendel's work, the fundamental principles of heredity were revealed. We now know that genes, carried on chromosomes, are the basic functional units of heredity, with the capability to be replicated, expressed, or mutated. Today, the postulates put forth by Mendel form the basis of classical or Mendelian genetics. Not all traits are transmitted from parents to offspring according to Mendelian genetics, but Mendel's experiments do serve as an excellent starting point for thinking about inheritance. So let's dive into it. Gregor Mendel was a lifelong learner, teacher, scientist, and a man of faith. As a young adult, he joined the Augustinian Abbey of St. Thomas in Brno, in what is now the Czech Republic. Supported by the monastery, he taught physics, botany, and natural science courses at the secondary and university levels. And in 1856, he began a decade-long research pursuit involving inheritance patterns in honeybees and plants, ultimately settling on pea plants as his primary model system, keeping in mind that a model system is what we call a system with convenient characteristics used to study a specific biological phenomenon to be applied to other systems. Now, in 1865, Mendel presented the results of his experiments with nearly 30,000 pea plants to the local Natural History Society, and here he demonstrated that traits are transmitted from parents to offspring independently of other traits in dominant and recessive patterns. And we're going to dive into what that means in just a moment. But moving forward, in 1866, he published his work, Experiments in Plant Hybridization, in the Proceedings of the Natural History Society of Brno. Now, Mendel's work went virtually unnoticed by the scientific community, which believed, incorrectly, that the process of inheritance involved a blending of parental traits that produced an intermediate physical appearance in offspring. The blending theory of inheritance asserted that the original parental traits were lost or absorbed by the blending in the offspring. But we now know that this is not the case. This hypothetical process appeared to be correct because of what we know now as continuous variation. Now, continuous variation results from the action of many genes to determine a characteristic, like height in humans, right? Offspring appear to be a blend of their parents' traits. Instead of continuous characteristics, Mendel worked with traits that were inherited with distinct classes, specifically violet versus white flowers. This is referred to as discontinuous variation. Mendel's choice of these kinds of traits allowed him to experimentally see that the traits were not blended in the offspring, nor were they absorbed, but rather they kept their distinctness and could be passed on. Now, in 1868, Mendel became abbot of the monastery and exchanged his scientific pursuits for his pastoral duties. He was not recognized for his extraordinary scientific contributions during his lifetime. In fact, it wasn't until around 1900 that his work was rediscovered, reproduced, and revitalized by the scientists on the brink of discovering the chromosomal basis of heredity. So that's a bit about Mendel's history. Now let's talk about Mendel's model system. Mendel's seminal work was accomplished using the garden pea, or Pisum sativum, to study inheritance. This species naturally self-fertilizes, such that pollen encounters ova within individual flowers, and the flower petals remain sealed tightly until after pollination, preventing pollination from other plants. The result is highly inbred, or quote-unquote true breeding pea plants. These are plants that always produce offspring that look like the parent. And by experimenting with true breeding pea plants, Mendel avoided the appearance of unexpected traits in offspring that might occur if the plants were not true breeding. The garden pea also grows to maturity within one season, meaning that several generations could be evaluated over a relatively short time. And finally, large quantities of garden peas could be cultivated simultaneously, allowing Mendel to conclude that his results did not come about simply by chance. This is why the peas were Mendel's model system. Now, Mendelian crosses. Mendel performed hybridizations, which involve mating two true breeding individuals that have different traits. In the pea, which is naturally self-pollinating, this is done by manually transferring pollen from the anther of a mature pea plant of one variety to the stigma of a separate mature pea plant of the second variety. In pea plants, pollen carries the male gametes, or sperm, to the stigma, a sticky organ that traps pollen and allows the sperm to move down the pistil to the female gametes, or ova, below. To prevent the pea plant that was receiving pollen from self-fertilizing and confounding his results, Mendel painstakingly removed all of the anthers from the plant's flowers before they had a chance to mature. Now, plants used in first-generation crosses were called P0, or parental generation 1, or 0. After each cross, Mendel collected the seeds belonging to the P0 plants and grew them the following season. These offspring were called the F1 generation, or the first filial meaning offspring, daughter or son, generation. Once Mendel examined the characteristics in the F1 generation of plants, he allowed them to self-fertilize naturally. He then collected and grew the seeds from the F1 plants to produce the F2 plants, or the second filial generation. Mendel's experiments extended beyond the F2 generation to the F3 and F4 generations and so on. But it was the ratio of characteristics in the P0 to F1 to F2 generations that were the most intriguing and became the basis of Mendel's postulates. So now moving on to garden pea characteristics and what they revealed for the basics of heredity. In his 1865 publication, Mendel reported the results of his crosses involving 7 different characteristics, each with 2 contrasting traits. A trait is defined as a variation in the physical appearance of a heritable characteristic. The characteristics included plant height, seed texture, seed color, flower color, pea pod size, pea pod color, and flower position. For the characteristic of flower color, for example, the 2 contrasting traits were white versus violet. To fully examine each characteristic, Mendel generated large numbers of F1 and F2 plants, reporting results from 19,959 F2 plants alone. And his findings were consistent. What results did Mendel find in his crosses for flower color? First, Mendel confirmed that he had plants that bred true for white or violet flower color. Regardless of how many generations Mendel examined, all self-crossed offspring of parents with white flowers had white flowers, and all self-crossed offspring of parents with violet flowers had violet flowers. And in addition, Mendel confirmed that other than flower color, the pea plant were physically identical. Once these validations were complete, Mendel applied the pollen from a plant with violet flowers to the stigma of a plant with white flowers. After gathering and sowing the seeds that resulted from the cross, Mendel found that 100% of the F1 hybrid generation had violet flowers. Conventional wisdom at that time, right, with the blending theory, would have predicted the hybrid flowers to be pale violet for hybrid plants to have equal numbers of white and violet flowers. In other words, the contrasting parental traits were expected to blend in the offspring, but instead, Mendel's results demonstrated that the white flower trait in the F1 generation had completely disappeared. Importantly, Mendel did not stop his experiments there. He allowed the F1 plants to self-fertilize and found that of the F2 generation plants, 705 had violet flowers and 224 had white flowers. This was a ratio of 3.15 violet flowers per 1 white flower, or approximately 3 to 1. When Mendel transferred pollen from a plant with violet flowers to the stigma of a plant with white flowers and vice versa, he obtained about the same ratio, regardless of which parent, male or female, contributed which trait. This is called a reciprocal cross, a paired cross in which the respective traits of the male and female in one cross becomes the respective traits of the female and male in the other cross. For the other 6 characteristics that Mendel examined, the F1 and F2 generations behaved in the same way as they had for flower color. One of the 2 traits would disappear completely in the F1 generation, only to reappear in the F2 generation at a ratio of about 3 to 1. Now, upon compiling his results, for many thousands of plants, mind you, Mendel concluded that the characteristics could be divided into expressed and latent traits. He called these, respectively, dominant and recessive traits. Dominant traits are those that are inherited unchanged in the hybridization. Recessive traits became latent or disappear in the offspring of a hybridization. The recessive trait does, however, reappear in the progeny of the hybrid offspring. An example of a dominant trait is the violet flower trait. For this characteristic, flower color, white-colored flowers are a recessive trait. The fact that the recessive trait reappeared in the F2 generation meant that the traits remained separate, not blended, in the plants of the F1 generation. Mendel also proposed that plants possessed 2 copies of the trait for the flower color characteristic, and that each parent transmitted one of its copies to its offspring, where they came together. Moreover, the physical observation of a dominant trait could mean that the genetic composition of an organism included 2 dominant versions of the characteristic, or that it included 1 dominant and 1 recessive version. Conversely, the observation of a recessive trait meant that the organism lacked any dominant versions of this characteristic. So, why did Mendel repeatedly obtain 3-to-1 ratios in his crosses? To understand how Mendel deduced the basic mechanisms of inheritance that lead to such ratios, we must first review the laws of probability. So, probability basics. Probabilities are mathematical measures of likelihood. The empirical probability of an event is calculated by dividing the number of times the event occurs by the total number of opportunities for the event to occur, right? So it's kind of like a percentage, but you don't multiply by 100 at the end. It is also possible to calculate theoretical probabilities by dividing the number of times that an event is expected to occur by the number of times that it could occur. Empirical probabilities come from observations, like those of Mendel. Theoretical probabilities, on the other hand, come from knowing how the events are produced and assuming that the probabilities of individual outcomes are equal. A probability of 1 for some event indicates that it is guaranteed to occur, meaning there's a 100% likelihood of it occurring. Whereas a probability of 0 indicates that it is guaranteed not to occur, or 0% chance of occurring. An example of a genetic event is a round seed produced by a pea plant. In one experiment, Mendel demonstrated that the probability of the event, quote-unquote, round seed, occurring was 1, or 100%, in the F1 offspring of true breeding parents, one of which has round seeds and one of which has wrinkled seeds. When the F1 plants were subsequently self-crossed, the probability of any given F2 offspring having round seeds was now 3 out of 4. In other words, in a large population of F2 offspring chosen at random, 75% were expected to have round seeds, whereas 25% were expected to have wrinkled seeds. Using large numbers of crosses, Mendel was able to calculate probabilities and use these to predict the outcomes of other crosses. And this brings us to the product rule and sum rule. Mendel demonstrated that pea plants transmit characteristics as discrete units from parent to offspring. As will be discussed, Mendel also determined that different characteristics, like seed color and seed texture, were transmitted independently of one another and could be considered in separate probability analyses. For instance, performing a cross between a plant with green, wrinkled seeds and a plant with yellow, round seeds still produced offspring that had a 3-to-1 ratio of yellow to green seeds, ignoring seed texture, and a 3-to-1 ratio of wrinkled to round seeds, ignoring seed color. The characteristics of color and texture did not influence each other. The product rule of probability can be applied to this phenomenon of the independent transmission of characteristics. The product rule states that the probability of 2 independent events occurring together can be calculated by multiplying the individual possibilities or probabilities of each event occurring alone. To demonstrate the product rule, imagine that you are rolling a 6-sided die and flipping a penny at the same time. The die may roll any number from 1 to 6, whereas the penny may turn up heads or tails. The outcome of rolling the die has no effect on the outcome of flipping the penny and vice versa. There are 12 possible outcomes of this action, and each event is expected to occur with equal probability. Of the 12 possible outcomes, the die has a 2 out of 12, or 1 out of 6, probability of rolling a 2, and the penny has a 6 out of 12, or half, probability of coming up heads. By the product rule, the probability that you will obtain the combined outcome of rolling a 2 and flipping a heads is 1/6 times 1/2, or 1/12. Notice the word "and" in the description of the probability, though. The "and" is a signal to apply the product rule. For example, consider how the product rule is applied to the dihybrid cross. The probability of having both dominant traits, for example, yellow and round, in the F2 progeny is the product of the probabilities of having the dominant trait for each characteristic, which, when we discussed it before, we said that was 75%, which is 3 out of 4. So that would be 3 out of 4 times 3 out of 4, or 9/16. On the other hand, the sum rule of probability is applied when considering 2 mutually exclusive outcomes that can come about by more than one pathway. The sum rule states that the probability of the occurrence of one event or the other event of two mutually exclusive events is the sum of their individual probabilities. Notice the word or here, or in this description of the probability. The or indicates that you should apply the sum rule. In this case, let's imagine you are flipping a penny and a quarter. What is the probability of one coin coming up heads and one coin coming up tails? This outcome can be achieved by two cases. The penny may be heads, or the quarter may be tails. Or the quarter may be heads and the penny may be tails. Either case technically fulfills the outcome. By the sum rule, we calculate the probability of obtaining one head and one tails as multiplying the probability of a penny getting heads by a quarter getting tails, and then adding that to the probability of a quarter getting heads times a penny getting tails, which would be equal to 1/2 times 1/2 plus 1/2 times 1/2, which in this instance happens to equal 1/2. You should probably also notice that we use the product rule to calculate the probability of a penny getting heads and a quarter getting tails, and also the probability of a penny getting tails and a quarter getting heads before we summed them, right? Operations, order of operations. Again, the sum rule can be applied to show the probability of having at least one dominant trait in the F2 generation of a dihybrid cross. So as a recap, for the product rule, for independent events A and B, the probability of them both occurring, meaning A and B, is the probability of A times the probability of B. And for the sum rule, for mutually exclusive events A and B, the probability that at least one occurs, so A or B, is the probability of A plus the probability of B. To use probability laws in practice, we must work with large sample sizes because small sample sizes are prone to deviations due to just random chance. The large quantities of pea plants that Mendel examined allowed him to calculate the probabilities of the traits appearing in his F2 generation. As you will learn, this discovery meant that when parental traits were known, the offspring's traits could be predicted accurately even before fertilization.